Now we will study the solid phase immunoassay. It is also the viral diagnostic procedures and how we can use these procedures. The solid phase immunoassay is most common used test. Why? Because it is very cheap. It is not such a complex like the other diagnostic procedures like ELISA or the serological test. It is very cheap and very simple, and it can be used for both the composition and antigen detection. It can be used for the detection of the antigen, that how much antigen is present and what is the composition of the antigen and what kind of the antigen is present, the solid phase immunoassay. So what are the diagnostic procedures on the basis of which these solid phase immunoassays are used? This is the one of them is the ELISA, enzyme linked immunosorbent assay. Then ELISA, enzyme linked immunospot, we can use it, and then GCSPR, the grating coupled surface plasmon resonance, and then the solid phase radioimmunoassay. There are 4 kinds of the immunoassays which we can use for the diagnostic procedures. The applications of these procedures, these are highly sensitive for the measuring of the different kind of the antigen. What kind of the antigen? Even not only we can measure the antigen, but we can see the composition of the antigen, and antigens homogeneous simple such as purified proteins in the buffer, and as well as unidentified heterogeneous biological samples we can also see. The examples for the biological samples maybe include the cell lysates, tissue culture supernatant, bloods, and many other clinical samples which are isolated from the virus infected cells and which can be used for the diagnostic purpose.
Many simple rapid diagnostic tests detect specific antigens in biological samples by using an enzyme immunoassay. The purpose of this animation is to explain how a prototype example of this assay works. The enzyme immunoassay can be done in a multi-well microtiter plate or on any other solid adherent surface. I will use the microtiter plate in this example, so let's take a closer look at one of the wells of this plate to see what happens during the performance of the assay. The plate is prepared to perform a particular assay by coating the wells with antibodies that bind to the antigen of interest. Then the wells are filled with the clinical sample, which could be a sample of serum, respiratory secretions, cerebrospinal fluid, urine, or some other body fluid. If the antigen is present in the sample, it will bind to the fixed antibodies. In this example, the green shape represents the antigen of interest, and the other shapes represent other molecules in the sample. However, note that only the specific, or green antigen, and none of the irrelevant molecules bind to the antibody-coated wells. This accounts for the specificity of the test. The wells are then washed out to remove any of the unattached molecules, leaving the antigen of interest stuck to the wells. Now, a second antibody, directed against another epitope on the target antigen, is added. These antibodies are conjugated covalently to an enzyme, indicated by the yellow circle at the Fc portion of the second antibody. They bind to the antigen that's fixed in the well, and this provides a second level of specificity for the assay. The wells are washed again to remove any unbound antibodies, and in the final step, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate and the captured enzyme generates visible color in the solution. At the macroscopic level, the development of color indicates those samples that have second antibody bound to the target antigen in the wells. Thus, the wells that change color are the ones that contain the antigen of interest. In other words, the positive tests.
Now you can see the little video and you can see the details how the immunoassay can be demonstrated.